All right. Hi, my name's Aaron. I'm a postdoc here at Imperial College London in the group of Riley Green. And today I'll be talking about mapping nerve regeneration into free muscle grafts, and specifically how I guess the new Mica platform has been integrated into our workflow, and how it's been useful to understand some of these biological processes. So first off, kind of the context that we're working in, limb loss or amputation is generally not seen as an ideal endpoint for a patient. They typically go home with severe implications for their quality of life. And this kind of, well, huge ramifications for how they experience just day-to-day activities that we take for granted. When a amputation occurs, what happens is you have a lot of loss of tissue, but in terms of the actual nervous system, there is enormous remodeling that occurs just proximal to the amputation site. And this is because nerves typically innervate all of these muscles and, I guess, skin and other sensory organs that are present in that tissue that is removed. And this has to do something during that remodeling and recovery process. And so what we see in a lot of patients is that the end of the nerve where it is transected actually grows in and forms a tissue structure called the neuroma, where the nerve fibers from your motor sensory or motor fibers potentially innervate sensory fibers. This sets up a kind of pathological cascade of events where you might get aberrant sensation or even pain because you're getting crosstalk between these two normally independent nervous systems. So, one of the things that the surgeons and plastic surgeons predominantly have looked at is how can you engineer that nerve stump to be less problematic for the patient, giving them back a degree of, I guess, normal sensation is the goal. But what they're providing here is a kind of free tissue interface through which that nerve can reestablish a semi-real or semi-normal innervation pattern. So this is done by taking a piece of muscle tissue, also usually from the, I guess, amputated piece of tissue, limb, and then embedding the nerve into that defined, now de-innervated piece of muscle, suturing it all up together, and then allowing the whole tissue construct to recover over a period of time. Now, because nerve and muscle, the nerve normally innervates the muscle, these tissues have a preference for each other. So the nerve will preferentially re-innervate the muscle rather than talking to itself. And so in doing so, you reduce a lot of these aberrant sensations that lead to neuropathic pain and phantom limb sensation. And so this has been quite interesting from the perspective of engineers and technologists as a potential interface through which you can talk directly to the peripheral nervous system and also the central nervous system. So this is work that's come out of Michigan, where they've used this kind of tissue construct with a very simple type of electrode to power a neuroprosthetic hand, where you can generate complex movements and some sort of feedback with a very simple interface. And so what we were wondering is, can you take that surgical construct and potentially design a more sophisticated engineering solution for interfacing with that, which would potentially allow you to get perhaps more functions or more lifelike sensation? So in our experimental model for this kind of tissue construct, we're working with rodents and basically looking at a specific branch of the sciatic nerve known as the common peroneal. So this is the nerve that normally innervates the extensor digitorum longus, which is responsible for extension of the lower limb, specifically the toes. And if you transect this nerve and then remove the target muscle, the EDL, the extensor digitorum longus, from its host site, you can then reinnervate that muscle with its parent nerve in a situation that would be analogous to a surgical RPNI construct in patients. And so what people have done so far is looked at the degree of recovery that you get from this and whether or not you have any reinnervation. And that's kind of where we come in. So when we were curious about the level of reinnervation in this specific model for potential spatial selectivity from this construct in a kind of biohybrid interface, what we found was that the reinnervation of that muscle tissue doesn't come solely from the nerve that we use to embed it. So that is to say that our construct doesn't get one source of reinnervation. There's enough tissue regrowth going on that we seem to be getting innervation from other sources. So what you can see here is that depending on which branch of the sciatic nerve we cut, we're getting different degrees of, I guess, contribution to a compound motor action potential. And I guess we've discriminated this into different levels of innervation coming from each of these potential sources. So we wanted to study this, and one of the ways in which you can do that is to take the whole tissue, embed it, stain it, and try to figure out what's going on. So our standard protocol until now for understanding reinnervation of tissues was to take something called Lugol solution, which is an iodine stain. It gives a little bit of differential contrast for nervous tissue. Tends to stain mostly glycogen though, but we can still see our nerves. And unfortunately, it's not selective. So in the CT, what you can hopefully appreciate, is that you get a lot of regeneration of tissue that is very complex and non-discriminative. When we look at individual slice sections of this micro-CT, we can see the point of entry of our common perineal nerve into our muscle belly, but we can't really distinguish what's happening at that regenerative interface. The staining is not selective, and apart from being able to see that there is something there where the tissue is less organized, there's not much information on how new axons are forming, how they are branching, and whether or not electrical stimulation or the general health of these axon branches for the purposes of a future kind of bioelectronic interface. So we went to histology. This gave us a little bit more of a view of what we could potentially achieve. Here we're looking at a standard H&E stain, and you can start to appreciate that you see levels of nucleation or nuclear proliferation in the region where the nerve is embedded. This image was taken using the Leica system, Microsystems. I think it took about 10 seconds to acquire across the whole image, which is quite fast. It's automatically stitched, and allows us to get a very good holistic view of the kinds of tissue regeneration that we hope to see with whole, I guess, tissue architecture preserved. We can go into very high magnifications. I think this is a 63x zoom, local in the interface point between where we're getting our potential regeneration and our kind of post-striated muscle belly. And one of the nice things about this is that you have an automated water immersion system. So you don't have to move your sample, you get great spatial registration, and you know where you are. So it's not only useful for histology, but you can also potentially use it for fluorescence microscopy. And depending on the way that you do your staining, overlay your samples. This is not an overlaid sample, but this is the same kind of sample preparation, but with immunofluorescence for neurofilament to identify where we have our axons. S100B, which we were using as a marker for potential myelination, provided by Schwann cells in the peripheral nervous system, and a general nuclear stain just to get a good sense of the whole tissue architecture. What you can start to appreciate is that with the stain, we start to see and identify axonal projections deep within the muscle tissue. We're still lacking in the identification of the whole sense of where this axonal projection is originating from and where it's going. But it is, work in progress. So we've also tried to push the system in what we can do with 3D imaging and volume imaging. So we've taken the whole tissue and cleared it using the cubic protocol, applied our standard immunofluorescent labeling. And what we can start to see is that you get good preservation of the whole tissue, you get the striations of the muscle cells, and you can start to see where a nerve is going into a muscle right at this point here. And then you can also look at potentially or a little bit of volume measurement in these tissue samples up to about, let's say, 500, 600 microns, depending on the optical clarity of your tissue and the strength of the stain that you're interested in. It's not going to be the same as a dedicated piece of instrumentation for volume imaging, but it is a very good and easy first step, or at least we found it to be. So I just want to leave off by saying that so far the Leica Microsystem has had a very rapid learning curve. It's kind of my instrument of choice for training new people who need to do some microscopy but don't want to spend the time learning how to optimize a confocal setup. I've talked to clinicians who are doing research projects and they found it very easy to both learn the histo side of things, but also IF. There's easy ROI selection, great integration across whole areas. And the auto measure means that you can go straight from a whole tissue to a very specific ROI and still know where you are in your sample. It's also got great integrated general computational clearing algorithms, so you don't need to know too much about your sample in terms of your point spread function, also for your microscope setup, very easily between wide field and confocal mode. We've also started to use this for visualizing things like drug release and diffusion extent in other applications. And so it's been a very versatile, well, at least we found it to be very versatile and easy to use tool. So with that, I'd like to acknowledge the Green Group, most of the people who supported this project, especially Zach, who's our surgeon/PhD student, developing the RPNI and its corresponding technological interface, and also the Leica team for helping out with the hub and continued support. Thank you very much.